friends, welcome back to my YouTube channel Astin Padasale. So in this video we are going to discuss the previous year question that was asked from Immunology. Okay, so these questions I have taken both from your GATE Excel question paper and GATE PT question paper. So both group of students will be getting benefited. First question is idiotypic determinants of an antibody are associated with. See, idiotypic determinants of antibody are associated with your variable region. So for example, clue word is that idiots will vary from one location to another location. You just have this clue word with you. Whereas your isotypic determinants and your allotypic determinants are associated with the constant region of your antibody. Next question. Identification of blood group involved. So identification of blood groups involves agglutination reaction. Okay, I think you all had done this blood grouping in your labs. Might be whether you are a zoology student or biotech students. You have done this experiment. Correct? So identification of blood groups involve agglutination. So you need to know the difference between precipitation and agglutination. So if you take agglutination, it is a reaction between your antibody and an insoluble antigen. In other words, it can say insoluble antigen as a particulate antigen. If blood group antigens will be coming under the category of insoluble antigen, which is another other name for insoluble antigen, it's a particulate antigen. So agglutination means you need to remember the word particulate or insoluble antigen, whereas precipitation is a reaction between your antibody and a soluble antigen. Okay, the best example of precipitation reaction is your immunodiffusion. Rotoloni double immunodiffusion and your radial immunodiffusion, all the things will be coming under the category of precipitation reaction. In the precipitation reaction, antigen will be soluble, whereas in agglutination reaction, it is insoluble. Next, B lymphocytes originate from bone marrow, whereas T lymphocyte. So T lymphocytes also originate from bone marrow only. The only difference is that T lymphocyte, after originating from the bone marrow, it need to travel to the thymus for their complete development. Whereas B lymphocyte, they originate from the bone marrow and they also develop in the bone marrow itself. So you need to read the question correctly, because many students will be losing the mark by doing these kind of silly mistakes. That's what I'm telling you. Next, humanized antibodies, one in which. So humanized antibodies are one in which the CDR will be taken from mouse. So CDR, the full form is complementary determining region. In other words, it is called as hypervariable region. Okay, so those regions will be taken from mouse and it will be inserted into the remaining portion of the human antibody. Okay, so CDR from mouse and rest of the thing will be taken from human. So there is a difference between humanized antibody and chimeric antibody. So you should not confuse between humanized antibody and a chimeric antibody. Okay, so CDRs will be taken from mouse and it will be spliced into the constant region of human antibody. Next, nude mice. The nude mice refers to mice without thymus. Those nude mice will be having few genetic mutations that will not allow them to develop thymus completely. So they don't have thymus. If there is no thymus, ultimately the mice will not have T cell. There is no T cell and ultimately there will be no immune response at all. That's why nude mice are highly used in research purposes. Next, heat inactivation of serum is done to inactivate complement protein. Serum is taken from blood only. So there will be few complement protein is there. So we need to inactivate. So before that, serum is generally employed for growing your animal cells in animal cell culture. Okay. So since the serum is taken from blood, it will be having few complement proteins with them. We need to inactivate those complement protein. The complement protein is not inactivated means it will be leading to the process of complement mediated cell lysis and ultimately it will be completely destroying your cells that you are growing in the lab. And also second important role is that serum is heat inactivated in order to destroy mycoplasma. So it prevents the cells from mycoplasma contamination. For both purposes, serum is heat inactivated. Next, members of antibody protein family that have common structural features is collectively known as immunoglobulins. So immunoglobulin superfamily, they have a common structural family. Okay, so that's why IgG, IgM, IgA are called as isotypic determinants. Okay. Next, in ABO blood group system, antigenic determinants. So specifically, they are asking about ABO blood group system. Antigenic determinants are carbohydrate. If you take A blood group person, they will be having N-acetyl galactosamine and B blood group person, they will be having galactosamine. Okay. So if you take RH blood group, the antigenic determinants are protein. So okay, so if they had given some other option like glycoprotein, you can also select glycoproteins also. Next, a neonatally thymectomized mouse immunized with protein antigen shows. So the mice is thymectomized, so it doesn't have thymus at all. So ultimately, it will not show any response to antigen. So option D is correct. Next, lymphocyte interact with foreign antigen. In generally, lymphocyte will be interacting with foreign antigen only in your primary lymphoid organs, and there are two primary lymphoid organs. One is lymph node, and another one is spleen. So lymphocyte interact with foreign antigen in your lymph node. So D option is absolutely correct. Interferon beta is produced by. So interferon beta is produced by your virus infected cells. Okay. Next question. Hybridoma technology is used to produce, ultimately hybridoma technology is for monoclonal antibodies. So I think many of you will be correctly find out this particular answer. Hybridoma technology is only used to produce monoclonal antibody. Okay. The antibody which has specificity towards one single epitope of an antigen is called as monoclonal antibody. Next is again a question, recent question, and it is not a repeated question. Dendritic cells are involved in cross presentation of antigen. Kindly note the point. Sometimes they will be asking this sentence alone in an option they will be giving. Okay, so dendritic cells are the cells that is involved in cross presentation of antigen, and they are asking which protein is required for cross presentation. So generally it is an MSQ type of question. The correct answer is option A. So basic leucine zipper ATF like transcription factor, in short form it is called as BATF3 is alone needed for cross presentation. Rest of the thing are not at all required. So correct answer is option P is the correct answer. Next is which of the following is required for the development of B cells in bone marrow? The correct answer is option A. Yes, stromal cells are needed because stromal cells they will be secrete a cytokine called as IL-7. That particular IL-7 is required for your B cell development. It is required for all, almost it is needed for development of your B cell, maturation of your B cells and differentiation of your B cell. Mainly these stromal cell will produce a cytokine like IL-7 in order to control the size of your B cell. Okay, so stromal cell they will be acting as a support for your B cell development. Rest all cells are not required for B cell. And also, next question in ELISA, which of the following enzymes are conjugated to antibodies for detection of the analyte? You can simply eliminate Q. That is, trypsinase is not required for your ELISA and amylase is not required. So, by eliminating these two options, the correct answer is P and R. So, option A is correct, which means only two enzymes are needed. One is alkaline phosphatase, another one is horseradish peroxidase. Next question is then match the following type of question. Match the immunologic immune tolerance given over here with the respective outcome. So first is energy. Energy means unresponsive to antigen due to lack of co-stimulatory molecule. Okay, energy is opposite to energy. So we all have notes for gate examination. At one point what will be lethargic and will not be stating anything. So that particular state is called as energy. So energy means it is an unresponsive to antigen. Why energy condition is created means if the T cell doesn't hold any co-stimulatory molecule means then at that time point the antigen the body will become unresponsive. It will not react to the particular antigen. Next is activation induced cell death. So here also you can able to find activation word. Here also you can able to find activation. So Q you can match with 1. The activation induced cell death is generally employed to eliminate the activated T cell after the antigenic clearance. Okay. Receptor editing. So receptor editing is used to eliminate autoreactive B cell. So it will be giving a second chance in order to eliminate autoreactive B cell. Next is your regulatory T cell. In short form, it is called as T regulate T REG like that you can tell. So this T regulator, what is the role of this regulatory T cell means they will be inhibiting the autoreactive T cell at the periphery itself. Okay, so at the periphery itself, this regulatory T cell will inhibit the T cell that are autoreactive towards the host. Next question, match the cell type with their function. So in these days, there were many immunological questions are asked specifically for gate BT. I'm talking about gate BT and also that is match the following. So if you cover only 50% of immunology concepts alone means you can easily score the correct marks. Even if you followed this video completely means you can definitely, I think you can score for 50% of question that is asked from immunology, you can give the correct answer. So study, you should be highly motivated while studying for GATE examination, because main aim for publishing these kind of videos itself, many of you will not even touch the syllabus. So by posting these kind of revision series, you can know something about the concepts. And in GATE examination, questions do repeat. So that's why I've taken the past year question paper. So coming back to the question, see, B cells are involved in your humoral immunity. And your neutrophils, you need to remember phagocytes, and your T cells are involved in cytotoxicity. So your CD8 T cells, or otherwise it is called as cytotoxic T cells, and your mast cells are involved in your histamine associated allergy. These mast cells, they have granules. Those granules are filled with allergy. Those granules are filled with histamine, and this histamine will be released at the time of allergy. And that's the reason why you're getting itching, or you will be getting vomited due to food allergies. Okay. Mast cells are associated with histamine mediated allergy. Next is the hypersensitivity reaction and the corresponding condition. Type 1 hypersensitivity you can quote with host reaction to bee venom. And type 2 is involved in erythroblastosis vitalis. And type 3 is associated with systemic lupus erythematosus. And type 4 is associated with the tuberculin reaction. So the next, this question. So let me tell you few other examples also. So this hypersensitivity is very important, friends. So type 1 means you need to remember the word allergy. Type 2 means it is antibody mediator. Type 3 means it is immune complex mediator, and type 4 is delayed type hypersensitivity. So in type 1 IgE antibody are involved. In type 2 IgG and IgM are involved. Next you need to know the typical manifestation. Like for example, if you take IgE, you need to remember the word anaphylactic reaction, hay fever, asthma, food allergy, beehives. That's what the question was asked about, bee venom, and eczema. Okay, these will be coming under type 1. The example for type 2 mediated hypersensitivity reaction include your blood transfusion reaction and your erythroblastosis vitalis and your autoimmune hemolytic anemia. Type 3 reaction example include Arthur's reaction, serum sickness, glomerulonephritis, your rheumatoid arthritis. So this question will be definitely asked, so please pay enough attention. And your systemic lupus erythematosus will also be coming under type 3. And type 4 example include your graft rejection, tubercular lesions, and your contact dermatitis will be coming under type 4. Next is again an autoimmune diseases and corresponding organ affected. So Hashimoto disease is associated with the thyroid gland, and juvenile diabetes is associated with the pancreas, and multiple sclerosis is associated with brain, where the autoantibodies will be attacking the myelin sheath of your nerve. So it will be affecting both your brain and the spinal cord. And myasthenia gravis is associated with the skeletal muscle. So this myasthenia gravis autoantibody will be attacking the neuromuscular junction. Okay, so here you can be able to find the other example. Like for example, if you take this multiple sclerosis, the target gene or target organ is your brain. Whereas if you take this again, if you take the spontaneous infertility and myocardial infarction and myasthenia gravis, and you need to know about the disease and whether in which or which part, what is a self antigen or what is a target organ. This is the most recent question, I think so, friends. Okay. Next, which one of the following is not a part of human non-specific defense system? There are two different systems. One is specific and another one is non-specific. Non-specific defense will be coming under innate immunity, and specific defense will be coming under adaptive immunity. The example of innate immunity, or in other ways non-specific defense, include your barriers, skin, mucus, saliva, all those things. Your phagocytes, pattern recognition molecule, TLR, complement interferon will be coming under this non-specific defense. Okay, whereas your T lymphocyte, B lymphocyte includes both your plasma cells and memory cell, and antibodies will be coming under this specific defense. So here your interferon, mucus, saliva will be coming under non-specific defense, and your antibody, they doesn't belong to non-specific defense system, they belong to a acquired, like adaptive immunity. Okay, the correct answer for this question is option D. Next is with regarding to the experiment. Which one of the following combination of following statement is true about antibody structure? So first statement is limited proteolysis of rabbit IgG with the enzyme pepsin. Pepsin generates two antigen binding region and an Fc fragment. So you look on this particular image. So papain digestion, it will be producing three fragment of equal molecular weight. There will be one Fab fragment and another Fab fragment and one Fc fragment. This Fab fragment, they have an univalent. They have an univalency. Okay, so there will be three fragments will be generated with regarding to papain digestion. So you are cutting papaya into three parts. One is for you, one is for your papa, another one is for your amma. Okay. Next, if you take your pepsin digestion, they are creating only two fragments. Main reason is that pepsin will be cutting below the disulfide bridge, whereas your papain will be cutting above the disulfide bridge. In other words, we can say papain will be cutting in the N-terminal portion, whereas your pepsin will be cutting in the C-terminal portion. So this pepsin will be producing two fragments. One fragment is a divalent fragment that is referred as Fab2. It will be consisting of two fragment, and these two fragments, Fab fragments, are joined by a disulfide bond, and it will be producing several Fc subfragments. Okay, so here Fc is intact. There is no breakage in Fc fragment. Here Fc fragment is subdivided and it will be producing many tiny Fc fragment. Okay. So here option B is incorrect because limited proteolysis of rabbit IgG with enzyme, papain will be generating two antigen binding regions. But here it is giving us pepsin. Okay, with regarding to option statement number 2, statement number Q, limited proteolysis of rabbit IgG with enzyme papain generates a single bivalent antigen. But no, your pepsin will be generating a single bivalent antigen binding site and peptide fragment. So papain will be generating a univalent fragment and your pepsin will be generating a bivalent fragment. So ultimately the correct answer is R and S. So option C is correct. Statement R is that Fc fragment of IgG can self-associate and crystallize into a lattice. So Fc means fragment crystallizable. Okay, so from the name only it got this particular statement. So it has a capacity to self-associate and they have a capacity to crystallize to form a lattice structure. If you take this particular Fab2 fragment, it is composed of both heavy chain and light chain. So if you look on this particular image, it is made up of heavy chain here and light chain also. Okay, so correct answer is option C. Next, which one of the following amino acid is catalyzed by activated macrophage to produce reactive nitrogen species? The correct answer is arginine. So don't collapse arginine with asparagine. So arginine is involved to produce reactive nitrogen species like your nitrogen dioxide, nitric oxide, nitrous oxide, all those things. Those will be coming under reactive nitrogen species. Okay, they are involved in the antimicrobial activity. Next, which of the following cytokines are endogenous pyrogen? The endogenous pyrogen are nothing but those substances which are coming from inside of our body and they are producing fever-like effect. And your interleukin alpha, interleukin 1, 6, 8, tumor necrosis factor, which is short form, it is called as TNF alpha, and your interferon, all will be considered as an endogenous pyrogen, in which IL-1 is a potent endogenous pyrogen. The correct answer is tumor necrosis factor and interleukin 1. This transforming growth factor and interleukin 10 are not an endogenous pyrogen. So option A is correct, P and Q is the correct answer. Next, which one of the following is not a principal component of innate immunity? Already we discussed. You consider innate immunity means mucosal epithelia, dendritic cell and complement system will be coming under innate immunity. Whereas your memory B cell, say both your memory B cell, T cell, antibody, plasma B cell, all will be coming under adaptive immunity. The correct answer is they are asking about which is not. So ultimately option D is the correct answer. Next, which one of the following mechanism is used by the human pathogen to evade host immune response? They are talking about a human pathogen. So the correct answer is antigenic variation. The best example is your coronavirus. They keep on changing their spike protein or few other protein present with them. Another example is your influenza. Hemophilus influenza, H1N1, about the bird flu, swine flu, all those things. It is two things are considered: antigenic drift and antigenic shift. Next, macrophage eliminate pathogenic bacteria upon activation by. Okay, so upon activation by your CD4 T helper cells. Okay, so macrophage, they are involved in eliminating your bacteria after it get activated by your T helper cells. The correct answer is option C, that is CD4 T helper cells. Here you need to know about another concept, like your macrophage is belonging to a innate immune cells, and whereas if your CD4 T cell is coming under an adaptive immune cell. So first this macrophage will activate themselves and then it will be activating your CD4 T helper cell. This CD4 T helper cell will help them to eliminate a particular pathogenic bacteria. So both the macrophages and CD4 T helper cell, they cooperatively engage to eliminate the pathogenic bacteria from the human body. Next, which one of the following condition promote the development of human autoimmune disorder? It's a very simple question. Inability to eliminate self-reactive lymphocytes is the first and foremost criteria to develop human autoimmune disorder. Second one is generation of autoantibodies. Okay, so option C is correct. Whereas ability to eliminate self-reactive T cell, induction of T regulatory cells, or they don't bring a condition of autoimmune disorder. So autoimmune disorder arises if there is any inability, if the body can't able to eliminate self-reactive lymphocyte, means they will be producing autoantibody against the host pathogen, host component. Okay, next. So this is a immunoglobulin with a match the following type of question. So IgG is present, it is an antibody that is present in highest concentration serum. So you can simply eliminate option B and option D. Considering option A and option C, IgA, IgA is a major antibody in external secretion like your bronchial mucus. So Q is 1. So you can eliminate option C. Correct answer is option A. IgE, it will be protecting us from parasites and helminthes infection. IgM is an antibody that appears first in the serum after exposure to an antigen. So you just remember, when you got your fever, means the first person who will be helping you is your mother. So M for mother. So IgM is the first antibody that appear first in the serum after exposure to the antigen. Next, the reaction between antibody and antigen that are detected by precipitate formed in an agar gel. So it is an example of, it is referred as your immunodiffusion assay. Okay. Next, based on heavy chain, which one of the following antibody has multiple subtype? It is your IgG and another one is IgA. So IgG have 4 subtype: IgG1, IgG2 and IgG3 and IgG4. Here I had given other important things. If you take your IgA, means it is having 2 subtype. One is IgA1 and another one is IgA2. Next, energy. Energy we already discussed. Energy refers to unresponsive to and responsiveness to antigen is called as energy. Next, production of monoclonal antibodies by hybridoma technology requires splenocytes. So splenocytes are those cells that are isolated from your spleen. So in a monoclonal antibody, two important players are your B cell and your myeloma cell. So B cells are taken from this spleen only. So that's why for monoclonal antibodies, the splenocytes are taken, and spleen is a primary lymphoid organ where your antibody will be encountering your antigen. So for isolation of your B cells, splenocyte is needed for your monoclonal antibody. Next is a match the following type of question. See, mast cell, they are providing the defense against helminthes infection. So P is 4. So you can simply eliminate C and D. Next, natural killer cell, they will be expressing CD56. So Q is 2. Okay, here's both option have Q as a 2. Next, neutrophils, they will be containing a azurophilic granules. So all your white blood cells, they will be granular, and this neutrophils is having a azurophilic granules. It is made up of various kind of antimicrobial substance. Okay. Next, your dendritic cells. Dendritic cells, they contain a long membranous projection, just like your dendrites. So the correct answer is option B is the correct answer. Next, type 2 hypersensitivity reaction is mainly mediated by. So it is mainly mediated by IgM and also it is mediated by IgG, either IgM or IgG. Next, membrane of mature B cell. Membrane of mature B cell is made up of IgM and IgD. So remember the word MD. The mature D cell have IgM and IgD. Next, pick the correctly matched pair. Okay, so the correctly matched pair are Q and R. Activated B cell alone has a capacity to undergo class switching, and your pre B cell, they are made up of surrogate light chain. So here if you take, first is pro, next is pre, next is immature, next is mature. Okay, if you take your pre B cell, they have surrogate light chain. Okay, so they have mu chain and surrogate light chain. Next, again a match the following type of question. Basophil, they hold a receptor for IgE. So they have a Fc receptor for IgE. Both your basophil and mast cell have the Fc receptor for IgE. Your T cell, that is your cytotoxic T cell, will produce this perforin. Next, your B cell is your plasma cell, and your neutrophils are involved in the process of phagocytosis. Okay. Next. Which of the immune cell listed below are agranular, which means they don't have granules with them. The correct answer for this question is your monocytes and T cell. R and S are the correct answer. Granular lymphocytes include neutrophils, basophils and eosinophils. Okay, and also you can include mast cell. Mast cell is also coming under granular. Mast cell have two important proteins in the granule. One is histamine and heparin. They are located as a granules inside this mast cell. A granular includes your B lymphocytes and T lymphocytes, and also you can add your monocyte. Next, which of the following statement about IgA is or correct? So first is IgA secreted in colostrum, correct. IgA is transported across the cell by transcytosis. It is also a correct one. IgA its secretion is facilitated by poly-IgG receptor. Yes, of course. Secretion is facilitated by poly-IgG receptor, which is located in your epithelial cell. The last one, it is primarily exists as dimer, absolutely, but in serum, they are generally present as a monomer. Only in few secretions, they will be existing as a dimer. So here they have given mostly IgA is present not in the serum, but in the secretions such as tears, saliva, sweat, mucus, they will be present as a dimer. Okay, so in serum, IgA will be present as a monomer because there is no secretory component with them. Whereas if you take secretory IgA, they are dimer. Okay, so here they are adding a word serum. So it primarily exists as a monomer in serum. So this option is absolutely incorrect. So option A, option B and option C are correct. It is a multiple select question. Next, again and match the following type of question. So mostly immune cells they are asking match the following type of questions only. NK cells, so NK cells they are involved in antibody dependent cell mediated cytotoxicity, and B cell they are engaged in antibody production, and your mast cells are involved in histamine production, and your neutrophils they are the first cell to be recruited at the site of infection. Okay, so they are the first cell to be recruited. Once a pathogen is coming inside of the body, they are the one who will be first reacting. The human major histocompatibility complex is polygenic and also they are polymorphic. Okay, so option B is correct. Polygenic means they are made up of more number of genes. Okay, and this polymorphic means they have more number of alleles. Okay, so human major in histocompatibility complex is polygenic and also at the same time they are polymorphic. They show enhanced polymorphism when you compare with some other genes. Binding of an antibody to its cognate antigen does not involve. So binding of an antigen to its cognate antigen involves only non-covalent interaction, which include electrostatic, van der Waals, hydrogen bond and ionic interaction. Here the question, binding of antibody to its cognate antigen does not involve covalent bond. Covalent bond is not required. We need only non-covalent bond. Next is again and match the following type of question. So yeah, integrin. So integrins are nothing binding of cell to endothelium. So integrins helps the cells to bind to endothelium. Okay. Next is microglial cell. They are phagocytotic cell present in the neural tissue. Next is TLR. TLR full form is toll-like receptor. Toll-like receptor 7, the ligand for this TLR7 is your single-stranded RNA. Okay, so it will be recognizing the single-stranded RNA coming from virus or bacteria. Dendritic cells. So dendritic cells are involved in, we already discussed, antigen processing by cross-presentation. Okay, so just read the question. Whenever you are working on PYQs, just read the question completely and also the answer. So here I had given the TLR and the microbial ligand and from which microbe they are isolated. See, TLR7 and TLR8 are involved in recognizing the single-stranded RNA from virus. Whereas if you take this TLR9, they are involved in recognizing this CpG unmethylated dinucleotide from your bacterial DNA. Okay, so just give a read on these things. Okay, so we'll be continuing other questions in the upcoming videos. So thank you friends, thanks for watching this video.